Hello everyone, I'm Shadi Darvish Shafighi. It's my pleasure to talk about TUMOROSCOPE, which is about inferring clonal evolution of tumors from spatial transcriptomics and bulk DNA sequencing data. Firstly, I want to talk about intra-tumor heterogeneity. As you know, cancer is an evolutionary process, and it can be started by one mutation, and it will gain a lot of mutations, and it will produce a lot of clones during its process. And this heterogeneity can introduce significant challenges in designing effective treatment strategies, and it correlates negatively with patient survival, which is so important for us, and we need a solution for that. And the solution so far is multi-region DNA sequencing, which is coarse-grained, and we don't know about the position of the clones in each bulk sample. So we need a model for high resolution localization of the clones, and here it comes our model TUMOROSCOPE. TUMOROSCOPE get the spatial transcriptomics data, and we use this to getting the read alternative and total read counts. Also, we use HNE10 images for detecting the cells in spots and to identifying the presence of the tumor or immune cells in the spots. We also use whole exome sequencing data to infer the evolutionary tree using existing methods, and using these and three types of data, we will infer the fraction of the clones in each spot using TUMOROSCOPE, which is the graphical model, probabilistic graphical model, and I will talk about it here. Um, as we know, the probabilistic graphical model have some variables, and you can see these variables here. Each node presents a variable, in each arrow present a conditional distribution, and the hidden variables are the unshaded nodes, and HSK is the most important hidden variable that we have because it's the fraction of the spots S coming from clone K. And we have also AIS and DIS, which are observed variables, and shaded observed variables are shown by shaded nodes. And AIS and DIS came from ST data, and they are alternative and total read counts. Also, we have CIK, which is the genotype, and FK, which is the discretized fraction of clones, and they came from whole exome sequencing data. We did the parameter estimation using Gibbs sampler and Metropolis Hastings inside Gibbs sampler, and we wanted to know how well the model inferred the composition of the clones in the spots. So we used 5 different configurations. First, the basic one, which has medium number of mutations in clone genotypes and medium number of clones in the spots. Then we increased and decreased this number of mutations in the clone genotypes and the number of clones in the spots, and we created these 5 configurations, and you can see the results here. And we run the model for 15 times on the simulated data, and for the in the left hand side we consider the number of cells as a hidden variable, and they are unknown, and in the right hand side the number of cells are given to the model and they're known. So you can see the error. We have some outliers, but you can see most of the times we have the error less than 0.02, and this is acceptable error actually. And we after that we applied on the real data. We have prostate cancer data from this paper that you can see here, and this data has 3 cancer sections, and each section have three different type of data that we need. uh, we use whole exome sequencing data to infer the phylogenetic tree using Canopy, and you can see the tree here with the corresponding mutations in each node. And then we infer the fraction of the clones in each spot. At the left hand side you can see the H&E images. The pathologist actually annotated the the cancerous part of the H&E images, and we use these parts and we only applied our model on these annotated parts, and we get these results. And on the left hand side, each spot is represented by a pie chart which is showing the composition of the clones in each spot. And we wanted to validate our results. We assumed that spots in the same neighborhood should have same clone compositions, and with these assumptions, we used correlation for validation. We had two sets of spots, four spots in adjacent spots, and we calculated the correlation in both of them. And you can see in the results here that the correlation of the adjacent spots are always much higher than the correlation in the far spots. And it can be seen also in the results here in the right hand side, because for example in this right side you can see the pink clone is dominant and in the middle part the green clone is dominant, and so on. At the end I want to say that this model is a step forward in understanding the spatial heterogeneity. It benefits from three types of data: spatial transcriptomics, H&E images, and whole exome sequencing data. We selected high quality somatic mutations and inferred the clones using Canopy. We matched the genotypes of the clones to the mutations found by ST data in the spots. We validated the results using comparison of the correlation of the adjacent and distant spots. And at the end, I want to thank my supervisor, Dr. Evosh Turek, also Professor Jens Lagergren for his huge helps, and my awesome collaborators and colleagues. And thank you for listening. I'd be happy to take any questions now.